Continuing the cell cycle, remember the big question, how can one cell become a multicellular organism? Remember the cell cycle has two main phases, interphase and mitotic phase. Interphase is what most of the cell is undergoing, most of its life. Some cells only go through interphase and never do go through the mitotic division or phase. In each phase, also make sure that you know that interphase is made up of 3 stages. The G1 is rapid growth and replication of organelles because it begins right after the new cell is created. So the new cell is created and has to undergo rapid growth and get all those organelles in there. Then goes through the S, which means synthesis, the S stage. And that's when the DNA is replicated, or the chromosomes are replicated. And then the last is the G2, which is preparing that cell to undergo cell division, which is the mitotic phase. The mitotic phase cell division is in 2 parts, mitosis and cytokinesis. Interphase is the period during the cell cycle of a cell's growth and development. Most cells are in the interphase. So when we look at the cells of an onion root tip under a microscope, you're going to see a lot of them that are interphase. And you have to hunt and look for the ones that have actually started the mitotic division. So there's rapid growth and replication or copying of the membrane-bound structure called organelles. So remember, replication of organelles is in the G1. Replication of the DNA, copying of the DNA, is in S. And then preparation for cell division is in the G2. So in the G1 phase, another thing that is important that's going on in there is the production of RNA. Remember what organelle produces RNA? The nucleolus. And what does the RNA, what does it have a role in? It's actually, some RNA makes up a ribosome. And a ribosome has the nickname protein factory because it is synthesizing proteins. So all of that is going on in the G1 phase. The S phase, synthesis, making something, and it's DNA replicating itself. Remember, DNA is what makes up chromosomes. So in actuality, it is a chromosome making an exact copy of itself. So in the human cell, we have 46 chromosomes in each nucleus. So if it is in the S phase, by the time the S phase is over with, how many chromosomes would be in the nucleus? Well, remember, they duplicate. They make a copy, a replica of themselves. So 46 times 2, that's your answer. Now, one new thing that we haven't seen before is, what are these duplicated chromosomes, this DNA that's been synthesized? What does it look like? So you have 2 identical chromosomes, and they are attached by a structure that is called a centromere. That whole thing, what is actually being made in the S phase, is a sister chromatid. Remember chromatid? That's the DNA inside the nucleus. So sister chromatids are the chromatin. So the chromatin duplicated. So if this were a human cell, we would have 46 of these sister chromatids. Whereas if you just counted each chromosome, you would have 92. So this is the replication of DNA. So it's the process, the replication of DNA that produces these sister chromatids, and it's in the S phase. In the gap phase, we're not going to spend any time on these checkpoints, but there's checkpoints that will not allow the cell cycle to continue unless the conditions have been met. And here's some more about the Check. So the mitotic phase is what we're going to study in depth in this section. So mitosis. Mitosis is basically, remember, the mitotic phase is two parts, mitosis and cytokinesis. Mitosis is basically making another nucleus that's exactly like the other one. That's the whole purpose of mitosis. And it has to go through stages in order to get this accomplished. So the equal distribution of the parent cell's genes between the two new daughter cells. The two new daughter cells are the end result when the mitotic phase is over. After cytokinesis, they're each called a daughter cell. And the key component of daughter cells is they have an exact copy of the DNA in each cell's nucleus. And that is to give them the whole genetic code. Each cell has to have that. We all began as one cell. And in that cell, human cells, mom gave you 23 chromosomes, dad gave you 23 chromosomes, and that gave you your genetic code. And it was in one cell. And now you are trillions of cells. And so that one copy, that first copy, had to be replicated over and over and over again every single time a new cell is made, no matter what kind of cell it is inside of you. So during mitosis, the contents of the nucleus is dividing to form two identical nuclei, one to go in each daughter cell. Like interphase, as I said, it goes through phases, through steps. You need to know these steps in order. Prophase, Metaphase, Anaphase, and Telophase. Some people say Telophase. So those are the phases of mitosis, and we're going to learn what happens in each phase. The longest phase is prophase. The shortest is metaphase. If you learn prophase real well, and you can just reverse it, then you will get what telophase is, because basically it's almost the reverse, the exact reverse of prophase. So in prophase, these are all the important things that you need to know that happen. It's the first phase of mitosis. There's a couple of things that disappear. The nuclear membrane makes sense on why it has to disappear. In order to form two nuclei, it can't be bound up by that nuclear membrane. It has to be able to divide into two. So it disappears. Why God chose it to have the nucleolus disappear, I don't know. But that disappears as well. Now there's something that's going to be needed in this process, and it forms in this first phase. And they're called spindle or spindle fibers. And that's how the chromosomes are going to travel and move inside that cell, by way of these spindle fibers. In an animal cell, they originate out of what we call the centrioles. In a plant cell, plant cells do not have the centrioles. Now these chromosomes, which remember they are sister chromatids, because they were formed in the S phase of interphase, they start changing shape. And they're still attached by that centromere. So they start coiling up, they start condensing. It kind of gives them an appearance of a big fat X. Here's a diagram showing you the chromosomes. Obviously these have not begun to coil yet, so they're not short fat X's, but you can see the centrioles with the spindle fiber forming. The centrioles actually duplicated, and one pair is going to rotate and move to the opposite side of the cell from the other pair. And then the nuclear membrane, you can see as the dotted line, is disappearing. Under the microscope, when we look at the onion root tips, this is what you will see, and you're looking at what's called early prophase, because if you look at it, you can still see the nucleolus in there. But whenever you have the nucleolus, but you start seeing individual chromosomes, and you won't see a whole sister chromatid, because this is like one slice of a cell. So you're getting some pieces of sister chromatids. And so you won't be able to see the big fat X, but you will see a bunch of scattered parts of the chromosomes. Metaphase, the shortest of each. The key is to remember metaphase middle. Those sister chromatids are moved to the middle of the cell by the spindle. And so they're pushed there. So they line up. All those little centromeres are lining up in the middle of the spindle, or what is known as the equatorial plane in some books. So in the middle of the spindle, we've got the sister chromatids all lined up. Why does God line them up? Because they got to split correctly. We have to have two identical cells with the whole copy of all the genes, all the chromosomes. So they line up first so they can separate properly and go to opposite sides and form new cells. Here's a drawing of seeing all the chromosomes, the sister chromatids all lined up in the middle of the spindle there. They call it a metaphase plate. I've also heard it called an equatorial plane. Here's what it will look like under the microscopes when we do lab. And you can see all the chromosomes there in the middle of the cell. Anaphase, think of anaphase as the separation phase. These sister chromatids have to separate now. So one of the chromosomes goes into one new nucleus, and the other exact chromosome, that copy, goes in the other nucleus. And so they cannot be called sister chromatids anymore, because a sister chromatid is two identical chromosomes attached by a centromere. They separate. So separated sister chromatids have a name now, and they call them daughter chromosomes. So anaphase begins as soon as the split takes place. As soon as daughter chromosomes are formed, that's the beginning of anaphase. And during the travel time, where it travels from the middle of the cell to the opposite end, which they call poles, they don't call them north and south because they don't necessarily face north and south, but they're poles. And so that period of migration, that period of separating and traveling to the poles, that is known as anaphase. As soon as they reach the poles, it will be telophase. Here's the artist's conception, and you can see them being dragged, actually being pulled by those spindle fibers to the opposite poles. Here's in the real microscope what it looks like, and you can see the evidence of them being pulled apart. And that's the clue to look for anaphase. Now, telophase, remember, begins as soon as those daughter chromosomes get to their poles. Now we have to have some nuclei form, so we have to have a nuclear membrane start to surround them. So that if you think of the prophase, what disappeared? The nuclear membrane and the nucleolus. So what has to reappear now? Nuclear membrane and nucleoli. And so that's some of the things that happen in telophase. Now remember what happened to the chromosomes. Those sister chromatids coiled up, became like fat X's. So now they have to do the opposite of that. They have to begin to uncoil. The spindle was formed in prophase. It is no longer needed now that it got those daughter chromosomes to the opposite poles. So since it's not needed, it will be broken down and disappears. And the end result is these two identical nuclei. And here we have the artist drawing. See all the pairs separated, and at the places where the centrioles are, see the nuclear membrane beginning to form, and you should see something else now. You should see where this is starting to pinch in the middle, where that division plate, not division plate, but that equatorial plane or metaphase plane is. Now if you think about in a cell, now we have the new nucleus, we have one for each cell. What else needs to divide? And that's the cytoplasm with all the organelles in it. So that process happens in telophase towards the end of it, and it is called cytokinesis. Here's the real picture under the microscope. This is a plant cell, so it doesn't pinch in. Think of why. Why can't that plant cell pinch in like the animal cell? Goes back to the boundaries we studied. The plant cell has a very rigid exterior boundary, an extra one, called the cell wall. So instead, it has to make this little division between the two nuclei, called a cell plate. Some books will refer to it as a division plate. And that's a dead giveaway under the microscope that this particular cell is in its last phases. It's in the telophase, and cytokinesis is going on. Now, some important things to realize is that the two cells that result from this mitotic division that we just looked at, from prophase all the way to telophase, they are called daughter cells. And as soon as they are created, they begin their life. And so they begin the cell cycle. And they obviously go into the G1 phase, which is rapid growth and the replicating of the organelles. So they have enough to be a functioning cell. And another key thing to know is their DNA, those genes, those chromosomes are identical to that beginning cell, which is referred to as the parent cell, and some books even refer to it as the mother cell. So where is the mother cell? Is it still there? Do we have 3 cells now? No. That mother cell now became the 2. And so it no longer exists, just the 2 daughter cells. Here's again in a nutshell, and I think these diagrams are in your book, of what happens in prophase. Copied DNA condenses into chromosomes. The nucleolus disappears, nuclear membrane disappears, spindle fibers form. Metaphase in the middle. Chromosomes line up in single file in the middle of the cell. Anaphase, they separate and travel. So sister chromatids separate, spindles begin to shorten, that's pulling them towards the opposite poles, and the cell begins to lengthen. And telophase is almost the opposite of prophase. Now we have to have the nuclear membrane forming, the chromosomes begin to unwind, uncoil, the spindle breaks down, no longer needed, and you've got 2 identical nuclei. Now we need the cytoplasm. Cytokinesis is, and you can get the connection, cytokinesis, cytoplasm. So the division of the cytoplasm is cytokinesis. And here you can see the division of the parent cell cytoplasm after mitosis is finished. That's cytokinesis. And what it's giving is each of those daughter cells, it's giving it some cytoplasm and organelles. Does it have to be exact like the nuclei? No, it doesn't have to be 50% and 50% where the same number of every organelle is in there. No. It could have 40% and 60%. Why? Because it's got the DNA and it's got everything it needs to make more organelles, because that's what it's doing in interphase. Now, you need to know a few differences between animal and plant cell division. First of all, it takes longer in an animal cell to take place. It still varies. Not every cell is going through this phase in the exact same time period. Plant cells do not have those centrioles. And in the cytokinesis, because the plant cell has the rugged, rigid boundary, it cannot pinch in, so it has a division plate, a cell plate. Whereas, because the animal cell just has that flexible and real pliable membrane, it can pinch in, which actually is called the cleavage furrow. Here's showing you the differences between the pinching in and the division plate or the cell plate. On the left there is the animal cell, and on the right is the plant cell, and you can see the differences. Now the main concept you need to understand here is some cells, when they undergo this whole cycle in cell division, they form 2 organisms. So any unicellular organism, such as what? We've studied some. Bacteria, algae, blue-green, protozoans such as amoeba, paramecium. When they go through cell division and form the 2 new daughter cells, it's actually 2 new amoebas. So from one, the parent, the mother amoeba, came 2. So it's duplicates, making another new organism. Therefore, it is reproduction. But in your body, let's say your skin cell is going through the end, cytokinesis, and forming the 2 new daughter cells. So we have 2 new skin cells now. Does that make another human being? No. So therefore, it's not form of reproduction. It's actually growth, producing more cells than are dying. And that's what's taking place when you are growing. Here's a good shot of what you'll see when we do the lab. It's not like when you look under the microscope, you're going to see all the phases in the right order. You're going to see one with prophase, and then right next to it's going to be metaphase, right next to it's anaphase, and so on. They're going to be scattered throughout, and a lot of them are going to be in interphase. So what do you look for? You look for the chromosomes. If the chromosomes are visible and scattered, it's prophase. If the chromosomes are lined up in the middle, it's metaphase. If the chromosomes are being pulled apart, it's anaphase. And if the chromosomes are forming 2 new nuclei, they're at opposite poles, that's telophase. And especially with a plant cell, you can look for the division plate, the cell plate. Now here, try to guess what these are. In the upper left-hand corner, what phase? And these are animal cells right here. Did you get anaphase? You got it correct. What about the lower right? Do you see any visible chromosomes like you do, those dark things? That is just a cell that's in interphase, going through its normal life. What about the upper left? See all those dark things in the middle? Those are the chromosomes. So they're in the middle, so that's metaphase. These are animal cells in the lower right. You see them how they're pinching in? You see the 2 new nuclei? That is telophase. Okay, in the upper left-hand corner, what phase? Metaphase is correct. Upper right-hand corner, prophase. It's actually early prophase because you can still see the nucleolus in there. The lower left-hand corner, you got it, telophase, because you got that cell plate and 2 nuclei. And the lower right-hand corner, anaphase, they're being pulled apart. Prophase. Upper left, anaphase. Lower right, interphase. Did you get that one right? Upper right, metaphase. But you can see an anaphase right over, a couple over. And then the lower left, you've got 2 daughter cells. Actually 2 cells in interphase. Daughter cells would be smaller. Okay, here you've got, in the upper left, you've got your 2 daughter cells side by side, the division plate, cell plates, now a cell wall. The upper right is prophase. The lower left, anaphase. The lower right, telophase. The upper right, prophase. The lower left, metaphase.